Hi, everyone. Could you take your seats, please? 2:26. So we're 1 minute over already. Welcome for a fantastic lightning talk session. I'm Kat Gold. I work at the Wellcome Trust in London. I'm very excited to chair both this session and the following plenary. We have a wonderful, diverse array of speakers. They're going to be talking to you for 5 minutes each on a fantastic array of different methods and systems. We're going to be very strict on timing, so it's 5 minutes each. And there probably isn't time for questions, so I would encourage you now to find them afterwards, express your enthusiasm proportionate to the color of their lanyard. And just a shout out to all the virtual attendants. Thank you. We know you're there. Please send through your questions and comments, and we'll make sure they get back to you. So without further ado, our first speaker, Jian.
Okay, thank you for giving me this opportunity to share some of the recent work that my lab is doing. And I'm a new assistant professor at Mass General Hospital and Harvard Medical School, so I will start with a thank you to all my wonderful co-mentors and collaborators and the funding agencies, and especially all the amazing team and the talented students who actually started to work with me, including Cecilia and Charles, who were used to work with Aviv. And so my lab is interested in developing new technologies to decode and engineer cell fates. So over the past few years, we developed a series of novel technologies to reconstruct developmental trajectories, including the Waddington OT, optimal transport work with Eric and Aviv, and also a CISO model for reprogramming, and more recently, the Raman to RNA, again, in collaboration with Aviv. So the central theme my lab is interested in, and many of the people in the audience are asking, is how can we define the cell states, types, and functions? So I think many people have their own favorite assays to define the cell states and types or functions. And some people are using imaging, some people are using genomics. And I think these two fundamental or orthogonal measurement methods are actually providing different information about the cell states, and then they both have pros and cons. For example, genomics, they are interpretable, but the fundamental limitation is that it's destructive. You have to kill the cells or lysis the cell to give the information, and also it's still very expensive with limited throughput. On the other hand, imaging can be very low cost, non-destructive, and high throughput, but the challenge is that the imaging features are really hard to interpret. So our solution is, can we actually combine the best of the two worlds and just to use the information that can be provided by each measurement and then link them together. So that's possible because of the recent technology advance in both machine learning, imaging, and single-cell genomics. So for example, that the ImageNet can help us to translate the imaging information into synsets or labels. And with the AlphaFold, you can translate the sequence into structures. And more recently, with DALI 2 from OpenAI, you can also translate text into images. So we wonder, can we translate the imaging information into single-cell multi-omics informations? So that became possible because of some of the recent advance in chemical images, chemical imaging methods that can generate more than 1,000 dimensions per data point. So it's a super high hyperspectral imaging method, and then we collaborated with a wonderful postdoc, Cecilia, in Aviv's lab, and also with Peter So and Tommaso to develop Raman to RNA. And we first test this in the mixture of iPS cells and fibroblasts and to see whether we can predict the gene expression. But what is the Raman microscopy that we're using? It's a chemical imaging method that can provide the composite measurement of all the molecular vibrational energies inside of a cell. So it's a composite measurement and gives you the biochemical phenotype of a cell, and it's high dimensional, up to 1,000 dimensions. So we first test this whether you can actually predict the marker genes from iPS cells and fibroblasts. The answer is yes, and then we apply this to the whole reprogramming process and see if we can actually link this Raman image data with the single-cell RNA-seq data generated during the Waddington OT work. And then the short answer is yes. If you wanted to know more how we can do this, just come to check our bioarchive or send me an email. So I just wanted to give you one highlight data from the Raman to RNA. So as you can see here, and you're probably very familiar with the UMAP. So the bluish is the reference single-cell RNA-seq, and then the yellowish dots are the actually single-cell RNA-seq data predicted from the Raman microscopy images. And you can see how if we can co-embed this two together and how wonderful they can align with each other. And beyond just the Raman microscopy, there are actually a large variety of other very cool imaging methods that's fairly unknown to the genomics or biology communities. So for example, some of the new imaging methods that we're testing that can generate this mouse brain map within 10 minutes. So if you're interested in how we can do this, just come to talk to us, and also shoot me an email or follow us on Twitter. So thank you so much.
Which is quite impressive. So please go and find him for questions afterwards. Next up, we have Cecilia from the Human Technopole, who's going to tell us about cross-tissue immune analysis.
Thank you. It's a great pleasure to be here and have the opportunity to talk to you today. So I just completed my postdoc in the Teichmann lab and just started my own lab in the Human Technopole. And today, I will tell you today about the work we did during my postdoc. This is not... Okay. In Sarah's lab in collaboration with the Jones lab in Cambridge University and with the Cambridge Repository of Translational Medicine, where we started asking the question of, can we improve our understanding of the spectrum of immune cell states that we find across human tissues that are usually not sampled when we look at human cells in peripheral blood, which is the most common sample that we can obtain, and really gain a better understanding of the diversity of immune states that are critical to determine the outcome in disease. And we are currently expanding this work with the CCI Immune Aging Network led by Nir Yosef in collaboration with Donna Farber and Peter Sims' labs. In this work, one of the key initial challenges was to perform a systematic annotation of cells across tissues, and to tackle this challenge, my colleague Chuan Xu and Tomas Gomez, who was a former PhD student in the lab, developed Cell Typist, which is a cell type classifier based on a broad collection of public data sets that we have curated to a very high degree and harmonized in terms of cell annotations, and have built a model to predict immune cell type identity. At the moment, it's available online for use and also to download and use locally. And it's not just the immune model that we initially developed, but actually also a broad collection of human and mouse models focused on single tissues and also cross-tissue models. For today's talk, I will focus on some findings on the T-cell compartment. So in the tissueimmunecellatlas.org, you can find all data available to explore or download. And we divided the immune system in three major compartments and analyzed some features of adaptations across tissues. And some of the key findings that we have is that in both the CD4 and the CD8 and gamma delta compartment, we have very specific resident memory populations that are very restricted to epithelial tissues such as the gut. And one layer that we added for the T-cell compartment is that we could look at clonal relationships by establishing the T-cell receptor sequence and could find cross-tissue sharing across different locations, but also many expansions in the jejunum across the lamina propria and the epithelial layer. And what I find also particularly exciting is that we could find cells within the same clone that belong to different stages of memory T-cell differentiation. And in the future, we are performing ex vivo activation experiments to understand better the relationships between these cells, and also, as I mentioned before, with the whole CCI Immune Aging Consortium, extending the amount of samples we have, also to look at different age groups that have quite different structure. Just very briefly, in my lab, the plan is to focus on early life immunity, and in particular adaptive immunity, which is shaped dramatically in the first ages of life. And I'm currently building the team, so very happy to talk to anyone interested. And I'd like to thank the many collaborators that made the work possible, our funders, and also the donors and their families, and you for your attention.
Thank you. Christoph said I had to be a very Germanic chair, but so far I haven't needed to, so thank you. Next up, Anand from A-STAR and the Singapore Immunology Network.
Thanks for the introduction. First thing, I landed in Vienna, I was like, okay, did I go back to Singapore? The weather here is almost the same as Singapore, so if you haven't been to Singapore, you're not missing out much. Right, so I'm not a single-cell biologist, I'm more of a translational immunologist, so I'm going to give you questions so that you can help me answer. What we are looking at is primarily to understand allergies, especially Asian allergies, better. This figure shows there are ethnic differences, which we've discussed before, but also we don't have specific biomarkers, which makes treatment and also prevention difficult. So my lab focuses on using endotypes of AD, which are subdivided phenotypes to better diagnose and treatment patients. Right? So this was a paper we published last year, which in short showed that although atopic dermatitis skin, non-lesional skin, looks similar in patients, when you look at their microbiome composition, they are quite different. In fact, if you look at the diversity on the left, 2 AD patients or eczema patients could look very different. In fact, some patients look more like controls. And here we could show that not only is the microbiome different, the skin function is different, and more importantly, these patients also tended to have a microbial dysbiosis, which had more virulence. And surprisingly, when we looked at the plasma of these individuals, they also had a higher cytokine content and an inflammation profile. So this made us wonder, why would your skin disease be affected by the immune system, or is the immune system affecting the skin disease? So then we went into single cell, we took PBMCs from the patients, and we looked at their immune profiling by looking at the different clusters. The one on the left is the T cells, the one on the top right is the myeloid cells, and one on the bottom are the B cells. So surprisingly, similar to the plasma profiling, we saw that although these two groups of patients both are eczema patients, they tend to look quite different. And in fact, this ADA, which was the milder form of eczema, had a profile almost which looked like the healthy patients. And when we went in closer to look at them, we could actually show that these people who had this dermotype is a skin endotype, they had a particular subset of inflammation which was different from the dermotype A. And if you look at the right, I've given the 2 types of cells, which cluster with the cluster which is enriched in these dermotype B patients, which tends to be more inflammatory monocytes, which could be why they have a severe form of disease, which could also be why they have a higher infectivity rate. And surprisingly, in the AD dermotype A patients, they tend to have more of the classical monocytes, which don't secrete as much cytokines and probably don't have as strong an inflammatory profile. Lastly, we've found this paper published last year, which showed that when you treat monocytes with LPS, the single cell profile looked quite different from the baseline, which is what you would expect. But surprisingly, a lot of the genes that they discovered, because this was in a healthy cohort, a lot of the genes that they found was the similar genes that we found. So what we did was we went back to a new cohort and then recruited patients who were control in AD, and we could show that these patients had a higher inflammatory profile, and this specific subset of monocytes, which we had found in the single cell, we could also figure them out in flow. So now we are trying to stratify these patients based on their dermotype so that we can see whether the entire loop of the immune phenotype, the skin endotype, and the clinical phenotype helps us to define the patients without going through high-throughput single-cell profiling as well. So here we also show that the TNF-alpha monocytes, which is quite similar to what has been shown here, highlights an important network, which is the pro-inflammatory monocytes. And these are surprising because you don't need to go to the tissue as yet. We can actually get them from simple blood profiling. And as an immunologist, flow cytometry is quite routine for us, so it's a lot easier to do flow cytometry than single-cell profiling. So lastly, this work wouldn't have been possible without the people that were involved. So as I mentioned, The clinicians are on the left, the skin biologists are there, and the immunologists are here. I also started my lab last time, but I don't have a slide saying we are hiring, but I'm happy to talk about collaborations. And I thank you for your time.
Next up, we have Margot Amott from the Beth Israel Deaconess Medical Center.
Hi, thanks. I'm Margot. I'm a postdoc in Evan Rosen's lab at Beth Israel, and I'm really excited to talk to you guys about our efforts to create an atlas of white adipose tissue. So some of you might be wondering why adipose tissue. It's commonly thought as kind of a unique or an inert storage organ, but it actually has a unique capacity to continuously expand and remodel throughout adulthood. We know that it's associated with a number of metabolic diseases, and we know that as weight gain occurs and metabolic diseases develop, there's remodeling and an influx of numerous cell types within the adipose tissue that are thought to be involved with the development of disease, but are still poorly understood. So the second question some of you might have is what took so long? Some of the tissue networks here have had atlases for years at this point, and we've only published this year. And the short answer is that adipocytes themselves, the mature fat-storing cells, are very difficult to work with. They're very large and very fragile and can't be sequenced using traditional whole cell methods. They're too large to fit in microfluidic devices used for droplet-based methods, and they're also too large to be FACS sorted into a plate. So all of the work, most of the work to date has only focused on the non-adipocyte cell types within the adipose tissue, which have been some really great papers, and we've learned a lot. But what our lab was really interested in was specifically getting an atlas that included adipocytes so we could study them and study their interactions with other cell types. So we switched to doing single nuclear sequencing. This had a number of advantages. One was that we could use frozen tissue, and the other, of course, is that we were able to sequence adipocyte nuclei because they could, in fact, fit into whatever we wanted to sort them into. So, as many of you might know, however, the switched nuclear data did result in an increase in ambient RNA levels, which is a big problem for anybody working with nuclear data, because in creating the nuclear prep, the cells are lysed and the cytoplasmic RNA is present in the prep and contaminates the resulting cell reads. So one of the methods that we used in creating our initial atlas was to calculate the spliced and unspliced RNA contents with the idea that cytoplasmic RNA had a much higher percentage of spliced RNA compared to the nuclear RNA. So by looking at spliced percentage in cells or in clusters, we could determine if those clusters appear to be contaminated with ambient RNA. As you can see here, spliced percentage correlated with mitochondrial percentage, another marker of ambient RNA specifically, especially in nuclear preps. So what we ended up with was a single-cell atlas of white adipose tissue from humans. We do have some whole cell data here, but most of our data is single nucleus because, as you can see, we were able to pick up adipocytes, which were specifically something that we were very interested in. We were able to subcluster the adipocytes and identify a number of unique subclusters. We performed immunohistochemistry to validate that the marker genes for some of these clusters were staining individual adipocytes in the tissue, and we also performed some analysis looking at the markers of these clusters and tentatively associating them with disease types that we're really interested in looking into further. So what are our next steps? Well, right now, we've switched to hashing, which is really advantageous in multiple ways. One of which is that obviously we're able to do more samples at the same time, which is reducing our costs and increasing our N. But the other thing that we weren't really expecting is that part of the hashing workflow we're using involves FACS sorting the nuclei in order to specifically count the number of nuclei we're loading per sample. And that ended up really cleaning up our data from the ambient RNA. You can see two knee plots here. They're both the same tissue, they're both human subcutaneous adipose tissue. And knee plot aficionados can tell that the hash knee plot has a much sharper knee and a much lower ambient plateau compared to the non-hash. So anybody else who's dealing with this problem, we really recommend at least doing a flow sorting step, if not hashing itself, in order to clean up your nuclei. Our other next steps are expanding the Adipose Atlas. A couple of the groups in the Adipose Network, including ours, are now working on weight loss studies. We have adipose samples from individuals undergoing bariatric surgery, as well as at 1, 6, and 12-month follow-up appointments that we're sequencing now in order to look at longitudinal remodeling of the adipose tissue with weight loss. Also, a larger goal for the Adipose Network is sequencing fat depots. Unlike many of the other organs that you guys are working on, there are a lot of fat depots throughout the body, and a lot of them are differentially associated with things like the development of metabolic disease. And so we really want to get as many of these as possible, and we really want help with that. So this is my call for anybody who's interested to reach out and join the Adipose Atlas. We really, really want to work with as many people who are interested in fat as possible. We're also very interested in tissues that have adipocytes that may not be studying adipocytes otherwise, such as breast tissue and skin. If you are doing single nuclear data and you have adipocytes and you want to talk, please talk to us. We really, really want your data and to work with you. And with that, I'd like to thank everybody. Please join the Adipose Atlas.
Thank you, Margot. And last but certainly not least, we have, excuse me, Oz Götze from the Ludwigs-Maximilian-Universität.
Thank you very much. I'm glad to be here and great to see you. First of all, I would like to start. With the people who done the work. Peter and Katrina run this project with our partners, Martina Scheffer and Mika Simons, and with our funders. So my group is interested on aging, specifically brain aging, and aging is the common risk factor for all the chronic disorders. And it's a hard topic to make progress, and 60 years ago, Richard Feynman gave us this advice. If you want to make faster progress, do like us, use more math. It took 60 years, but thanks to the single-cell genomics, we start to use mathematics. And my lab's workflow goes like that. We do differential dissociation white versus gray matter. And using electron and light microscopy, we develop hypotheses, test them, and again design another experiment. And this loop leads to identification of several white matter aging-specific cellular states of microglia and oligodendrocytes. And we have built up a hypothesis. So aging-induced oligodendrocytes are shedding myelin, and microglia in white matter responds to that by converting into white matter-associated microglia and clearing that, which somewhat attracts CD8 T cells, which induce interferon response microglia and interferon response oligodendrocyte. We bravely hypothesize that it leads to oligodeath, and this is the trigger for many degenerative disorders. This would be testable, but will take 3 decades to test this. And looking back to the Richard Feynman again, if we wanted to speed up the process, just make the electron microscope 100 times better and look at the damn thing. That's his words. And we thought how we can achieve that, and we thought maybe we can actually combine spatial transcriptomic MERFISH data with the electron microscopy. To test this, we generate an injury by an injection on one side of the brain and cut the brain one section for the MERFISH and the other one for electron microscopy. MERFISH data was excellent. You see the red where the microglia responds to injury, and we got around from 3 animals around 400,000 cells, and they show here. The adjacent section went to our electron microscopy pipeline, and after data generated, we overlapped this both data sets and try to get to the both transcriptomic and morphological information. Using that data, first we focused the electron microscopy, and we single cell annotated each cell by going through the around 15 sections of this electron microscopic data set. And we could identify In the injurious site, lipid droplet enriched foamy microglia, T cells, around T cells, unknown C cells that our neuroanatomists could not annotate, astrocyte, homocytic, activated microglia, both lysosomal and non-lysosomal, and these were just looking to the morphology we could identify. Since we have the adjacent section, what we done next is we use the Murphy's data, and as you can see, the LAM, which we define it through the cholesterol pathways, microglia, perfectly overlaps with the foamy microglia, so that we could link these two identities. The surprising finding was these unknown cells in the electron microscopy were the interferon response microglia. As you can see, they both cluster around the T cells. And when we use the neighborhood analysis, we see that interferon response microglia themselves cluster close to themselves and T cells, while the foamy and the LAM cluster by themselves. And this is, we think this is a really cool technology. What we would like to go next is now we are having a 15 micrometer sections, and we would like to go thinner. It's possible to get 40 nanometer sections, so 50 nanometer sections even. So from a single cell, we can get 200 sections and 200 different omics from a single cell, theoretically. And also, if we can align this very closely, we could actually maybe infer data from different resolutions, from spatial to electron microscopy, electron to spatial. Thank you very much.
Thank you. I think we have time for maybe one question or two. Is there anything online, Sam? Do you want a mic?
Hello, yes, we had a question from Ramana Ibrahim from Queen Mary University. What software can be used to combine both whole exome sequencing and multiplexed imaging data? And I think that was for the first speaker.
For the first speaker.
Would you like me to read it again? Yes. What software can be used to combine both whole exome sequencing and multiplexed imaging data?
Oh, that's a great question. So I think there are a large variety of packages that you can use for integrating.
If you take your mask off.
Integrating imaging data with spatial transcriptomics data, but we use in-house developed pipelines. Some of these, autoencoder pipelines, I think you can somehow leverage those to integrate a large variety of different imaging modalities with different, spatial transcriptomics data. Thank you.
Thank you.
Thank you. Thank you. We might have time for one more question. Should we take one from the audience here? I misled you into thinking there wasn't time for any, so no. Nicolas?
How scalable is the electron microscopy from the last talk?
Sorry.
How scale?
How scalable?
So right now we could have a 9mm to 5mm section. This is quite a large section we could get, but that's the limitation of the atom ultrasectioning. So spatial electron microscopy for 5 to 3mm regions our standard equipment can do. And larger, we need to find solution to make atom sectioning much new. It's an engineering problem.
Thank you. Brilliant. Okay, I think we should move on for the sake of time. So can you join me all in thanking the amazing speakers one more time? They did the job of providing a kind of flash of inspiration post-lunch, so we're all energized now. Next, we have a smooth segue into plenary session 3. This is part 1, focused on multimodal profiling and applications. And our very first speaker is going to be Nikolaus Rajewski from the Max Delbruck Center for Molecular Medicine. And I just want to, again, highlight that our virtual audience participants, we can hear you, we hear your questions, so please join in as well. Nikolaus.
Thanks so much for having me, and excited to present really the completely unpublished data, also not available on bioRxiv. How do I advance this? Shit. Okay. All right, so this is a talk about a human lung cancer sample from a female patient. Was operated, early detection, but despite chemo, 3 months later with metastasis, extremely aggressive. As you of course know, so this talk is about microenvironment. So for non-small cell lung cancer, there's immune checkpoint inhibitor treatment, which only helps 20% of patients to respond. The idea is, of course, to inhibit that T cells are inhibited in targeting tumor cells and doing their job. And tumor cells are, of course, master architects of the tumor environment to promote their own growth, to evade the immune response and resistance to therapy as well. So what we did is, in collaboration with NanoString, the CosMx platform, is we took a paraffin-embedded sample and sectioned it at 5 micron distances and sent every 6 or so section to NanoString. One section we did H&E, you'll see that. I'll not talk about one section where we did 10x Visium. And what the NanoString technology did for us is a membrane antibody staining, so that you can segment cells into nuclei and cells. And then they did about, for 1,000 genes, a few hundred were chosen to specifically encode signal transduction important genes. 1,000 genes in 350,000 cells, so about 160 million transcripts at a resolution of 0.2 microns. So we have on average then, when we segment and assign transcripts to cells, about 200 transcripts per cell. So if you do the usual UMAP, you can see that there's clearly there are tumor cells in the upper left part, 5 and 4. And you can also already see some interesting things that you cannot see with just single cell dissociated sequencing. If you look at the fibroblast clusters 0 and 1, which are different in gene expression, they have a different cell size. And I think that's just conceptually really important. We can start to really connect morphology to molecular data. So we leveraged the enormous lung healthy cell type data that were presented here from all the colleagues. And you can see that you get quite beautiful mapping of basically all the cell types to our sample. You also see that the segmentation is not working really 100%, so you get some spread of cells where probably adjacent cells were lumped into one cell because the segmentation algorithm is not perfect. So we are working on this. It will be improved, but I don't think it matters for the story I'm telling here. There was a paper about 44 patients' lung cancer single cell sequencing, grouped into low risk, high risk, and so on. And very nicely you can see that our data map best to the high risk tumor data, as one should expect, but that is a totally independent control. So we also did a three-dimensional reconstruction. There's a video which should run, but it doesn't. Oh, it does. It's running, okay. I'm not going to go more into this, but the data are good enough. We developed software for this. Stefan Preibisch is a lead author on this, who is now at Janelia. We developed software to align the transcriptomics, spatial transcriptomics, and to do a three-dimensional analysis. This is open source software, which is merging open source libraries from the imaging field with spatial transcriptomics. But I want to now go in where the cell types map that we see in space. So of course here you see the airways, then you see the tumor cells, you see the innate immune system, you see the adaptive immune system, and then you see the stromal compartment. So really filling the slice, I think, quite nicely. And now compare this to the H&E, okay? And so I think what becomes really clear here is that this is a kind of, let me say it in a grandiose way, the pathology of the future, right? There's just so much more information now. And of course, what you can also do now is to use machine learning to link the morphology or everything you have in the H&E to the molecular data. And of course, there are already people underway doing it, but I think the data quality here really allows you to do this in a beautiful way. Let's explore cell neighborhoods. We looked here at each cell and made a radius of 50 microns. So you have an average about 30 cells in that radius. Typically about 8 different cell types, and now you count. So you get a matrix where for each cell the cell types are quantified. Now you can define a graph on this by using some software from ecology and geography. Is not really the point, but it gives you it has a statistical model for the co-occurrence or absence of cell types within the same compartment. And so you can now identify cliques of functional relationships, for example, airway or vessels, or the tumor bed, or the dendritic cell tertiary lymphoid structure. So you can now take these data so you do a UMAP now and clustering on these contents which are in radiuses of cells. Okay, so this is not gene expression per se, this is contents of cell types, what kind of mixture of cell types you have, and clustering in a UMAP. And then you can see how this maps to the spatial cut that was analyzed by nanostring. And what you can see is really, I think, very nicely is that these niches occur multiple times defined in the way I described, so as it should be. You have the airways, you have vessels, you have here the tumor bed, and you have the tertiary lymphoid structure. And if you look in more detail now into the tertiary lymphoid structure, let's do that. And we do a molecular analysis of what kind of inhibitory pathways are actually there, now defined really as receptor-ligand interactions in proximal cells, okay? Of course by RNA, but still. And what you can see is that the famous PD-1 is CTLA4 pathways, which everybody knows. This is Nobel Prize, so this is what's used are there. But then we also discover that a less studied inhibitory pathway, it is also in the literature, but much less studied, is very prominent in the sample. So this could maybe define a target for a combination therapy. So I want to stop here. I want to say that a few points. We studied here a paraffin-embedded tumor. This is important because, I mean, the FFPE samples are the clinical standard, and this is also how the best H&Es are being produced. So this is just really important in practice. At sub-micron resolution for about 1,000 genes, cell segmentation was good enough to study the tumor microenvironment at single cell resolution. I made the point that we can see already that cell morphology, for example, cell size of different fibroblast clusters relates to molecular features, and I think that is conceptually really important. Okay, so the morphology, also the mechano features of a tissue can be linked really to the molecular programs. I think also for the machine learners, this is extremely important. Spatial relationships can be quantified and converted into graphs. You can cluster on spatial relationships and define meaningful biological niches. And these niches inform you about which immune inhibitory pathways you may want to target. What I pointed out is that we highlight here the galectin-9 TIM3 axis. And I think the entire approach of the data suggests that we can move forward pathology and target identification and the molecular understanding in new ways. I want to acknowledge people. The main person driving this project is a MD who is doing now his PhD in my lab. This is Tancredi Pentimali. I think it's also the new generation of scientists which is very excited about these single-cell approaches to diseases, and I think it's a pleasure to experience this. And then a number of other colleagues in the lab supporting this. We are collaborating with the Fred Clauschen lab at the Charité in Munich, who are doing machine learning for pathology. And this was a lovely collaboration with the nanostring and the cosmics, which I think will become available later this year. So thank you very much for your attention.
Questions for Nikolaus? Fabian?
Hey, Nikolaus, impressive scale and resolution. I didn't quite understand the sort of the two levels of niches, and then I think you call it sort of tissue context or so that you define. Maybe you can just enlighten us again. In particular, can you, with this sort of summary statistic locally, can you do differential comparison as well, which is something that we typically for images we cannot do, but with some stats potentially we could?
Yeah, so again, the niches are defined by clustering in space where now you don't have gene expression anymore, but you have the counts of the cell types in a radius of 50 microns around each cell. There are some more technical details behind this really, but I think that's really the main point. So that defines the niches, and really the test that this is meaningful is when you map them then onto space, right? And then you see how they're organized in space, and that to us makes a lot of sense. I'm sure there are other ways to define niches. Other people here also have done different ways of doing this. It's not really the main point of my presentation. I'm just saying that there's one way to define niches which makes sense. I think that what is of course now coming is an analysis of the same niche, what the differences between different locations, right? So this all must come. It's clear this is just the beginning of this kind of journey.
Hey, Nikolaus, it was really nice. Quick question on the 3D side. So in the beginning you showed kind of the stitching and the, sorry, forgot about it, and the 3D reconstruction. But in the analysis that was done later, was it done just on a section, or was it done actually in 3D? Say the 50 micron neighborhood, is that a two-dimensional neighborhood, or it's actually a three-dimensional cube?
No, actually, so we're a little bit behind. I was hoping, so yeah, ideally that would be done in 3D. At this point, this is done in 2D.
That's
but
in 2D.
This is possible. So,
of course. And the other question is on the, say you chose a 50 micron there, have you seen whether choosing different length scale for these actually defines different styles of the niches?
Yeah, of course, of course. Okay, so if you make the radius too big, it's not meaningful. If you make it too small, it's also not meaningful. So the interesting regime lies in between, and the question is at which scale. Now, I'm not even sure if there's just, one.
No, I would expect there would be multiple scales. That's why I asked.
Actually, so I think there are multiple scales. We just show here that for choosing one, you can already interpret the biology. But I think the interesting thing in this tumor will be to exactly do this, to look at different scales, how they are connected, and that probably defines something, I think, important about how tumors, why tumors are tumors.
Thank you. So we've got time for one more question, which is online.
Hello. We've got a question from Mark Halushka from Johns Hopkins University, one of our virtual participants. How different do you think the understanding of the TMI is between 2D and 3D approaches? Does this have to be approached in 3D, or could reasonable parallels be made in 2 dimensions?
Yeah, I mean, time will tell. I think that the tumor is 3-dimensional, and I think there are aspects of this will be controlled by molecules, the 3-dimensionality, because this is how the biology has to function. So intuitively, I think, yes, we of course also see that there are differences between the slices. There's heterogeneity at different positions in the tumor. That's of course well known from a whole number of studies. So I think definitely one slice is not enough. And I just made an argument why I think the 3-dimensionality is actually really important.
Brilliant. Thank you. We can thank Nicolas again. And please continue the conversations in the coffee break. Next, we have a virtual speaker. We're going to be joined by Ponpan Matangkasombu Chupong from Mahidol University. Ponpan, are you there?
Thank the organizer for inviting me to give a talk today on Thailand's single-cell RNA-seq experience from dengue immune response to HCA Asia 2022. We started our single-cell journey in 2017 when Dr. Deransawan and I started collaboration with Sarah groups at Sanger, who tremendously helped us with technical know-how to set up the first single-cell genomics lab in Thailand at Mahidol. Thanks to a series of Newton funds given to both Dr. Radom and I, and also Sarah and her team, that enable us for the initiation of the collaboration, scientist exchange through research funding, and also thank to Mahidol University for instrumental support. In 2018, we ran our first single-cell experiments and data analysis at Mahidol, and later that year, we had a workshop bringing Sanger scientists to Bangkok to disseminate the knowledge to broader Thai scientific community with attendance over 100 people. At the same time, we started profiling immune cells from dengue patients, which I will share with you our pilot study results today. In 2020, we then went on to join HCA Asia in Asian Immune Diversity Atlas, or AIDA project, and we were actively involved in HCA Asia and will host HCA Asia annual meeting later this year in November. Hope you can join us. So let's start with immune response in dengue infection, the dynamic immune response to natural human dengue infection at single-cell resolution. Dengue disease is a global burden affecting tropical and subtropical areas, as you can see in red here in the map, and Thailand is here. It is caused by 4 serotypes of dengue virus transmitted by Aedes mosquito vector, and it infected almost 400 million infections per year, resulting in 100 million symptomatic cases. Yet there's no antiviral drug currently, and there's only one approved vaccine with quite limited efficacy and could induce adverse outcome in children and also dengue-naive people. So the better understanding of disease pathogenesis is needed. Clinical course of symptomatic infection is well defined. Patients usually present with high-grade fever for 2 to 7 days. We call this febrile phase. In this phase, patients with mild dengue fever and severe dengue hemorrhagic fever and dengue shock syndrome cannot be differentiated clinically. Uniquely, the fever will abruptly drop at the day we call day defervescence. Unlike most other viral infections, this day marked the beginning of the critical phase of 1 to 2 days, when some patients would develop plasma leakage. You can see by increase in hematocrit and bleeding, and some would develop shock and die. But if the patient survive, they will enter the recovery phase.
the ability to predict which day the fever will drop, so will help triage the patient to the right place at the right time for close monitoring and deliver proper fluid therapy in a timely manner. However, there's currently no such predictor available. Therefore, we are interested in profiling immunological dynamics at single cell in depth every day, leading to day differescence. We call day -1 for 1 day before and day -2 for 2 days before day differescence. So we aim to identify the predictor for disease progression, as well as to understand the highly complex dynamic immune response leading to critical phase. Under dengue-free cohort, we collected blood samples from well-defined dengue patients of varying severity, dengue fever and dengue hemorrhagic fever, at different time points during the infection. In this study, we selected PBMC samples from 2 patients, 1 DF and 1 DHF, at 4 time points, day -1, day -2, day differescence, as compared to 2-week follow-up and healthy control. We single-cell profile these PBMC with 3'10x genomics. Cell type annotation was performed based on known gene markers, and we identified all expected cell clusters as shown in this UMAP plot. And when we look at cell type abundance dynamics over time in both the F patient and DHF patient, we can see the most abundant cell types are as expected, which are T cells, and they seem to expand the most at day differescence, while the plasma cell and plasmablast in green here seems to expand the most at day -1. Looking at transcriptional dynamics of cell type specific immune pathways across time in each patient, there's a lot of information here, but let me give you some example. Type 1 interferon signaling pathways in response to virus seems to peak early at day -2 in most of the cell type, especially monocytes, while B-cell mediated immunity and humoral response mediated by immunoglobulin seems to peak at day -1 in B cells, plasma cell, plasmablast cluster. Focusing on T cells, we can further cluster them into several clusters: memory, naive, and various effector T cells. We can see distinct effector CD4, CD8 group 1, 2, and 3, and also innate-like T cells here. When looking at the dynamic across time, the proportion of effector CD4 in pink and effector CD8 group 2 in red here seems to be the most highly dynamic and expanded mostly during fever or febrile phase and differescence. While when we look at the naive and memory T cells, the proportion of these cells over time seems to be stable. Interestingly, both effector CD4 and CD8 group 2 seem to upregulate homing marker to the skin, SELP-LG gene that encodes CLA proteins at day -1 in comparison to 2-week follow-up and healthy control, suggesting that these cells might home to skin to the site of viral entry through mosquito bites. We validate this at the protein level by looking at the expression of CLA protein on both CD4 and CD8 T cells, which express CD69 and PD1 here, which resembling effector CD8 group 2 and effector CD4. And we can see that at day -1, CLA upregulate as compared to 2 weeks in both CD8 group 2 and CD4. We validate this with higher number of patient samples, with 20 more patients and over 40 samples. And we confirm that in both the CD8 and CD4 that express CD69 and PD1, we can see upregulation of CLA expression mostly at day -1 as compared to week 2 in both DF and DHF patients and in both CD8 and CD4 T cells. So in summary, time-course single-cell RNA-seq reviews immune response dynamics during acute dengue infection. And we can see rapid transcriptional switching at day -1 and different dynamics in immune pathways in various cell clusters. We also show skin homing signatures observed in specific T cells and antibody-secreting cells, plasma cell, plasmablast during febrile phase. And the expansion of skin homing marker in CD69 PD1 T cells at day -1 was confirmed in the validation group with facts. So we're currently analyzing a larger cohort of patients comparing between severity groups and also use immune profile with 5'10x to include TCR and BCR data that enable us to look at the clonalities of these T and B cells. And we also profile a patient with asymptomatic infection that could help us elucidate the protective immune response in dengue infection, and also look at dengue-specific T cells using dengue immunodominant epitope peptides loaded on HLA tetramer, and sort these tetramer T cells and profile with SMART-SIG2 protocol. For application, this dataset could also help leading to the development of molecular markers that could predict disease progression. For example, identify day -1 that would lead to a critical period, so that we can help better triage the patient to the right place and give fluid replacement in timely manner. Now let's move to IDA project, Asian Immune Diversity Atlas project, in which Jian will present in more detail. But in brief, in IDA, we aim to build an atlas of Asian immune cell types, state and characterize differences between 6 major Asian population groups, and currently expanding. So in the first phase, Thailand joined IDA in 2020, together with Cham from Singapore, Jay from Japan, and Muyang from Korea. And we started together with Partha from India. So in Thailand, in the phase 2, we also expand collaboration, not only in Bangkok, where we, both Rodom and I are based, and we also expand to the north, northeast, and southern part of Thailand. We also have other ongoing collaborative projects besides dengue and IDA, such as dental pulp with Anand, and cancer, organoid, drug discovery, allergic disease, ocular disease, and COVID vaccine. So what do we learn from the single-cell genomics experience in resource-limited settings, such as in Thailand? We have several challenges, such as limited funding, limited infrastructure support, and initially we're the only lab in Thailand, and the government funding itself gears more toward application research. But there comes opportunity. We have unique research questions, such as tropical disease, questions such as dengue that we address in the first place. And multidisciplinary collaboration is quite seamless, and we have several clinicians collaborators and other scientists collaborators. With the work, international and local collaboration make our work possible, and also funding with international funding and co-funding grant scheme between international funders and matching with Thai funders will help move our research forward. And importantly, the knowledge and technology transfer and exchange between collaborators and within HCA consortium, especially HCA Asia, is very crucial. Last but importantly, we would like to welcome you to join HCA Asia annual meeting in Bangkok on November 3rd and 4th this year as a hybrid meeting. So if you can come on site or online, we'd like to welcome you. And we aim to open for registration soon, but please save the date. So all of this work has been done in close collaboration with Dr. Domenguez. Since the beginning until these days, we collaborate in all the single-cell work. And I'd like to also thank Sarah and Christine and our group for helping us set up this single-cell genomics lab in Thailand, and also the dengue work. Chandrika and Ananya are first author of this dengue paper that I presented today, and Ananya is actually there at the meeting on site, and you can also ask her a question and talk to her about our work and representing our team. And I'd like to thank all my team in the lab, and also Varadom's team, Sarah's team, Varadon, who is a postdoc leading the analysis of the larger cohort, dengue cohort that I told you about, and now he's back from Sanger to Mahidol and become a PI now. I also like to thank Dengue Free Consortium for providing all the dengue samples, IDA collaborators, and all the funders. So our group at Mahidol is expanding with 4 PIs, awesome research assistants, and brilliant graduate students. With that, I'd like to end the presentation, and we'll be happy to take any question.
Thank you, Pompan. I think we're going to beam you up on screen now. Do we have questions? Got one over there.
Hi, Anand here from Singapore. What level of resolution do you have in terms of age, gender, and other risk factors which could influence the dengue severity?
So in this cohort, we actually use only 2 dengue patients, 1 DF and 1 DHF, and both are male adults and have dengue 4. So because of the small number of sample, we cannot analyze those statistics, but we're currently analyzing a larger cohort, and perhaps we should be able to present the result in HCA Asia in November. Thanks for the question.
Great. There's another question, I think.
Hi, this is really interesting. It's Bruce Arano from Cincinnati. Do you see similarities with long dengue versus long COVID? There are people with dengue that seem to, they have a persistent autoimmune and febrile state.
That's a very good question. But in this study, we only looked at the 2-week follow-up. But in clinical experience, I agree with you that a lot of dengue patients has kind of long dengue with autoimmune disease and some further complication, but we haven't analyzed those yet. It's a good question and good research question to work on. Thank you.
Any more questions here or online? Okay, that's brilliant. Thank you, Pompan. And actually, without wanting to embarrass her, Anunya, could you just stand up? Because Anunya from Mahidol is here, and you can find her in the coffee break if you want to chat more about the work. Okay, thank you very much, Pompan.
Thank you.
Okay, and on to our third speaker of Plenary 3, Xiao Wang from the Broad Institute and the Chemistry Department at MIT. She will also be joining us online.
Hello, everyone. I'm Xiao Wang from MIT Chemistry and the Broad Institute. I'm very happy to share with you our research in the past few years on multimodal profiling of cell types and states in situ. So the first question is, why do we need a multimodal profiling of biological tissues? Taking brain as an example, single-cell sequencing has revolutionized the way we define cell types, and now we can actually have a very accurate molecular definitions for all the cell types. And from another angle, brain can also be characterized by its structures at different levels. And then thirdly, we can also mapping the brain through functional perspectives, such as calcium imaging and electrical recording and tissue histopathology. So with all these different angles of brain study, it's actually important that we can correlate all 3 of this type of information in one animal so that we can better correlate genes with functions. So with the unique development of spatial transcriptomics, now we can actually not only measure molecular informations at the single cell resolution, we can also have additional spatial information so that we can again convert the measurements of transcriptomics as a format of images, which give us additional opportunities to correlate the structures and functions. And ultimately, based on this highly multimodal integration of information, we may be able to develop more precise manipulation tools both for fundamental studies and also for therapeutics. So our lab has been working on this domain, which starts to from the development of StarMap back in 2018, where we combined in situ sequencing with tissue hydrogel clearing, followed by multiple mechanisms of error reduction to enable very high accurate RNA mapping of more than 1,000 genes simultaneously in intact mouse brain tissue, where we can achieve 3D cell typing. And in the past few years, we also developed a computational pipeline because it has been extremely challenging to analyze RNA expression in 3D. So ClusterMap is a hierarchical clustering computational pipeline where we consider RNA identity and density to identify subcellular structures and also to achieve single cell segmentation and tissue region segmentation. So combine these tools, we now have a very powerful tools to discover molecular cell types and space and states in space. And the very first question we want to address is, do molecularly defined cell types and tissues actually reflect brain function and dysfunction? So to this end, the very first example we explored is to combine in vivo calcium imaging as a readout of neuron activity and to correlate that activity pattern with molecularly defined cell types. To this end, we just combine calcium, in vivo calcium recording, and subject the animal into visual cued rewarding behavior. And through this process, we first identified 3 major cell types marked by TH, TAC1, and CHAT1 genes. And secondly, we found very clear functional segregation regarding these 3 cell populations, where one of them is corresponding to the predictive visual cues, where the other 2 cell types is corresponding to the actual consumption of rewards. So we do, at least in this case, confirm that molecular cell types reflect functional organizations of brain circuits. And secondly, we try to take a further dive into cell states where we combined electrical recording with 3D star map. In this particular case, we collaborated with Dr. Jian at Harvard, and we tried to use the development of soft electronics to embed in developing tissues such as human iPS cell-derived organoids. And then we critically designed a fluorescent electronic barcode where we can first record the living electrical physiology in living tissues, and secondly, we can imaging the fluorescent barcode to register the position of the electrode with the actual electrical channels, and thirdly, we can conduct in situ transcriptomics to profile the RNA expression near the electrode. Through this process, we try to correlate electrophysiology and gene expression at the single cell resolution. And through this type of cross-model analysis, we can identify electrophysiology-related genes programs that are responsible for the maturation of electrophysiology features during the organoid's development. And based on this type of gene to function correlation, we can also further adopt a machine learning-based approach to predict RNA expression from electrophysiology and vice versa. And lastly, again, it's still centering around the cell states, but we want to better understand what happened in the disease, in brain disease, for example, the neurodegeneration disease of Alzheimer's disease. So Alzheimer's disease has like 2 protein-based disease hallmarks. One is the extracellular aggregation of amyloid beta. The other is intracellular aggregation of neuron fibrillary tangles. So with the development of StarMap Plus, we can achieve integrated mapping of RNA and protein in the same samples. So through this approach, we can precisely dissect what are the cell types and what are the gene programs that are spatially associated with this disease hallmarks. And through this process, we can identify the cell state transitions and putting these type of transitions in the context of space to identify disease-associated spatial DEGs, which can be extremely powerful to study disease with significant morphological changes. So through all these 3 examples, we enabled better correlation or registration between molecular and functional states. Next, we want to, again, use the spatial transcriptomics as a way to evaluate potential gene perturbation and manipulation tools. So AAV is a widely used research tool. It is a single-stranded DNA virus. It's actually super important for all types of genetic manipulations of neuroscience, and also it's the most promising safe viral carriers for gene therapies. And many of the AAV variants actually have a different preference for tissue types and cell types, which people call as tropism. So a recently developed version of AAV PHP-EB allows for brain-wide transfection of transgenic constructs. The foundation of these tools is its capability to cross the blood-brain barrier. So in order to better profile the AAV tropism in the whole mouse brain, we engineered highly expressed circular RNA barcodes, and then through AAV registration, we designed a integrated StarMap Plus experiments where we can both mapping the circular RNA barcodes encoded by AAV together with endogenous genes to define molecular cell types. As a result, we first generated extremely high resolution molecular cell types and the molecular tissue regions of the whole mouse brain. And then we can precisely quantify the transduction efficiency of AAV across all the molecular cell types and tissues of the mouse brain, which will be a very valuable resource for people to evaluate the cell type of preference of AAV as a guidance for the experimental design. And lastly, I also want to introduce a new tool developed in our lab that further enable investigation of translational control in space. So spatial transcriptomics are important and critical to understand the molecular cell types and states. However, RNA may not be sufficient. While spatial proteomics is still very challenging, we devised an approach that we can profile protein synthesis in space, which we termed as RiboMap for spatially resolved single-cell translatomics in 3D. So the principle of RiboMap is that it can selectively amplify ribosome bound potentially translating mRNAs inside the tissues and combined with in situ sequencing for highly multiplexed readout at a high spatial resolution in 3D. And if an RNA is not bound by mRNA, there will be no signal. So as a preliminary data, we first applied RiboMap in mouse brain tissue. What we found is that we can precisely define molecular cell types using single-cell translatomics, where those cell types are actually aligned very well with the single-cell transcriptomics. But what's more is that we can directly visualize mRNA translation in tissues, which is actually very important mechanisms to shape cell structure and tissue structure. For example, here it's a RiboMap image of the hippocampus. And you know, many of these brain cells, such as neurons, astrocytes, and oligodendrocytes, they all have very branched structures. And there are many studies showing that there are both soma localized translation, which means translation is happening inside cell bodies, and also processes localized translation, where the translation happens at the periphery of the cells for specialized cellular functions. And through our study, we can discover many of those processes, localized genes, and to better understand what are the foundations of localized translation and what are the potential functional relevance for such type of decentralized gene regulation control at the translational level. So in collection, although I may have given too many examples, but the central message here is that 3D transcriptomics is not only just a standalone tools to generating spatial atlas, it also has many exciting opportunities to be integrated with other type of approaches to accelerate the discovery of gene to functions. Thanks everyone. I want to thank everyone in my lab and all my collaborators and funding agencies. I'm happy to answer any questions.
Thank you.
Michelle will join us. Thank you. That was an amazing array of methods in combination and by themselves. Do we have any question? Anyone else other than Nikolaus has already asked a question? There we go. Okay. Chair's prerogative.
Hi, thank you. It was a great talk. So, quick question regarding the multimodal recording with the electrical activity. I think there could be two ways of analyzing or looking at these results. On one side, you can look at the gene expression to identify the cells with a certain electrical activity, and then maybe you can look if a certain electrical activity has an downstream effect on gene expression. So I'm curious to know in which timing, what was the time in which you recorded the activity and then you look at the gene expression, and which of these two aspects you were aiming mostly to look at? Sorry, it was a bit long.
Thanks, that's a great question. So in our study, because we only used the electric recording as a recording tool instead of manipulation tools, and the biological system we are using is organoids, which is we only recording spontaneous happen like electrophysiology. So in our particular case, I think our goal is more like identify gene expression programs that are responsible for a emergent particular evolution of electrophysiology features. So it's more about the better understand the molecular foundation of electrophysiology. And because this development is a relatively slow process, like over multiple weeks, and we are sampling the time points at a resolution about like every week, so that I agree, I think most of the times when we looking at the development tissues, there may be some time delay between like what's happening molecularly and what's happening collectively at the tissue function level. But because we have sampled in a way that we ensure this, we give it enough time window between the electrical measurements and the molecular measurements. So I think at least in our case, these two steps are synchronized. And in the future for in vivo applications, I agree, like I think most of the times maybe if the developmental process is extremely fast, there may be some time delay. And for the particular study, for example, identify cell types in animal behaviors in the brain, I think our major purpose is like identify the cells responsible for particular neural activities, where we kind of like correlate stable cell types instead of those like very dynamic gene expression programs. But I think the time delay or the temporal resolution to enable such correlation is a very critical parameter to consider.
Thank you. Any final questions? If not, please join me in thanking all of the speakers for an absolutely fantastic session.
Thank you.
And I think your instructions are to go, get caffeinated, and be back here by 3:40 sharp.
Ask questions. Hi, everyone. Is it on? Hi, everyone. We're going to start our second half of the plenary session number 3, multimodal profiling and its applications. And we're really excited about this session. We'll hear about new technologies and computational methods, and we'll have a lot of exciting talks. And our first talk will be by Alexander van Udernaden, and he's from the Hubert Institute. And he's going to join us remotely. So Alexander. Alexander, we can't hear you.
Accessibility of the DNA using an enormous number of cells, basically, as I said, using commercial equipment. However you think about it, most of these technologies focus on really the first part of the central dogma. What happens after the mRNA is much more difficult to quantify by sequencing. So we started thinking, can we contribute a single-cell sequencing technique that allows you to look at the process of translation? Now, we started with ribosome profiling technology that was introduced by Jonathan Weissman now more than 10 years ago, which is a very elegant idea where you basically use the ribosome that are still attached to the mRNA to make a digest, then the RNA enzymes that digest the RNA and basically leave short fragments of ribosome-protected transcripts behind. Now, then you can sequence those little pieces, and then you know basically where the ribosomes were at the moment that you lysed cells. Now, typically, you would need millions of cells to actually do this experiment, so we had to think how to make this technology more sensitive. Now, a little bit about ribosome profiling. Now, when you do ribosome profiling experiment, not surprisingly, most of the reads that you will sequence will map to the coding sequence, because that's where most of the ribosomes are. And you'll find this very interesting periodicity of trinucleotide periodicity. And this is related to the fact that the ribosomes, of course, move in steps along the transcript, not as like at a constant velocity. And that, therefore, you will see preferred frame. Mike van Insberg, at the postdoc in our lab, really spearheaded this, both at the experimental side and the computational side. What he did is he sorted single cells into 384 well plates, then uses mNAs to digest away the RNA, which is not protected by the ribosomes, then get rid of the ribosomes and sequence those little footprints that are about 35 nucleotides in length. We do all the regular single cell barcoding, but we also do an additional size selection step, because as I said, these fragments, the footprints, have a very well-defined size. And this is the raw data. What you can see here are about 1,500 single cells, 2 different human cell lines. Each row is an individual cell, and the columns are positions along the open reading frame that has been aligned with respect to the start codon or the stop codon. Now, what you immediately see are those vertical lines, which are separated by 3 nucleotides. So that's what I showed you before. That's what you would like to see. This is the fact that the ribosome moves in steps. Also, you see a much reduced level of fragments upstream and downstream of the start and the stop codon. So this is also good. Now, I'm just showing you here an example of single cells looking along a single gene, actually. So this is a histone gene, and I'm looking here along the open reading frame of that gene and plotting the density of reads that we find. And here we have a population of cells that have been either starved in arginine or leucine, or they have not been starved. So the blue cells are the ones that are starved in arginine, and what you see is that these cells actually stall at a very particular position in the open reading frame of this histone gene, and this is actually very close to a region where there's a lot of CGCs. So there are 3 CGCs here. Now, CGC is indeed a codon that is encoding for arginine, so the cells here have a hard time going through that kind of roadblock because there is not enough amino acid available. So this shows actually that we can look at pausing or slowing down of the ribosome along the level of a single gene in single cells. Now, the rest of the talk I really want to focus on new things, because this was already published. What we really would like to do is to really basically have an assay on single cell level that can tell us, is a process regulated at the transcriptional or translational level? Now, suppose it's regulated exclusively at the transcriptional level, then, for example, you get more transcripts, but the number of ribosomes per transcript is not changed. The alternative would be that you actually keep the number of transcripts fixed, but you load more ribosomes per transcript. Now, currently, if you would only measure number of ribosome protected fragments, you would not see any difference. There are 4 here and there are 4 and here. So there's basically no difference. So what you need to do is you need to integrate the ribosome protected fragment information with the level of the transcript. So you have to integrate a single cell RNA-seq experiment with a single cell ribo-seq experiment. Now that's exactly what we do. We take basically one population of cells and then we sample 50% for a single cell RNA-seq experiment, the other 50% for a single cell ribo-seq experiment, and then we computationally integrate those 2 data sets, and then at the cluster level we can compare the levels of ribo-seq to RNA-seq. Now this allows us to get an estimate for the translation efficiency in those particular clusters. Now I'm going to show you an example here. So this is now an integrated data set where we integrate cell-seq2, which is RNA-seq, with the ribo-seq along the mammalian cell cycle. So this kind of, you see this circular pattern here where you go from G1 to S, G to M. So this is a beautiful UMAP that shows the cell cycle. And what you can do now is you can start defining individual neighborhoods in this UMAP. And in each neighborhood now you will have cells coming from the RNA-seq experiment and also cells coming from the ribo-seq experiment. And then you will be able to look at the scaling of those 2 experiments, basically measure how much they are different, and then allows you to, for each neighborhood, to get an estimate of the translation efficiency. Now this is an example of a gene. It's called TPT1, tumor protein translationally controlled. So this is almost a positive control. And at the level of the transcripts, you can see it's not changing much, but at the level of the ribosome protected fragments, I see quite a big change. So now what you can do is we run this through our models and we can extract the translation efficiency from this. And as expected from the raw data, you would see that the translation efficiency is high in mitosis and it's low in G0 and in G1. Now this is an example of a gene that does exactly the opposite. So it's very lowly translated in mitosis and it has a high translation efficiency in the G1. Now you can of course do this for all genes that you're interested in, all genes that you can detect. And that's what we do in this heat map here, where individual rows are genes, individual columns are cells that are sorted by basically the stage of the cell cycle. And you see, what we see is we see 2 major groups that I want to focus on. Group 1, which are genes that are repressed in G1, the TPT1 example that I showed you. Also, very interestingly, almost all the ribosomal subunits are in this group. And then in group 2 is the opposite behavior, it's anticorrelated with group 1, and it's repressed in mitosis. Now let's ignore the other groups for now, because I think group 1 and 2 are the most interesting ones. And what we can do now is we can start to look at the sequence surrounding the transcription start sites, for example, and they have a very different motif. So cluster 1 is CU rich, whereas cluster 2 has either a G or A at the beginning. So there's already a very strong difference in the transcription start site sequence. Now let's analyze this cluster 2 a little bit more. So these are the genes that are repressed in mitosis. Do they have any other properties that are different? Now what we did is we took the UTRs of those genes. So in blue here are the mitodically repressed genes, the cluster I'm interested in. And I look at the UTR sequence, either 5' or 3', and I let Vienna RNA fold this in the computer and determine or estimate the delta G. And what you can see is that this particular 5' UTR is actually pretty stable compared to the other clusters or other genes, basically. And the 3' UTR we don't see a major difference, but in the 5' UTR we see that this mitodically repressed genes, our cluster 2, is having more stable structures than the rest of the genes have. Now what's also interesting is that we find that if you look at the number of reads in the different parts of the transcript, the 5' UTR coding sequence and the 3' UTR, is we find that for cluster 2, the mitodically repressed genes, you find more reads in the 5' UTR than for the other clusters. Now this suggests that there are some more ribosomes perhaps in the 5' UTR. Now last thing I want to show you is that we also did a sequence analysis of all the sequences in the 5' UTR of this mitodically repressed cluster, and we got a very, very strong motif hit here, which is basically a repeat of CGGs. Now this is known to form so-called G quadruplexes, so it's basically a knot in the RNA, 5' UTR, and what we think is that this might be actually the cause for the repression of translation. We have to do much more work on this. I just want to share you some of the data that we have right now and show you where we go in the future, but we're quite excited by this. So let me conclude. We developed technology to perform ribosome profiling in single cells. I showed you that you can actually look at pausing at the level of a single gene in single cells. And this has been published, so you can look all the details up. But what we're really focusing on right now is to really try to figure out how to get translation efficiencies, basically estimated from this type of experiments. And the early data shows that we can actually predict very much based on the 5' UTR if a gene will be repressed or not in mitosis. And we try to figure out how to manipulate the cells so we better understand how this G quadruplex motif has biological importance. So let me thank the lab, a fantastic lab that has no fear. The single cell ribosome profiling was, of course, a huge risk. It's, most people would say it would never work, but I'm very happy that the lab is, fearless, basically. And I also want to thank the funding agencies for paying all the bills. Thank you very much.
Questions for Alexander? Alexander, what scale can you do the SC ribose-seq on?
Can you say again, which?
What scale can you do the SC ribose-seq on? How many cells?
How many cells? So this is, we do it based on 384 well plates, so I guess as many as you can process in a week, which probably is like about 20 to 30. So you can generate about 10,000 cells per week.
Great. I think Klaus has a question.
Yeah, thanks for letting me. Just a joke. Yeah, what can I do? I find this just very exciting.
Totally fine.
Alex, I'm just wondering how quantitative are really the data you're getting? I mean, are they quantitative enough to allow you modeling of rates, for example? So what kind of error bars do you think you have?
Yeah, good question. So we use exactly the same technology as with single-cell RNA-seq. So we use UMIs, and so we make sure that we can count. Of course, I think the major variability is not so much technical, but is biological between cells. So I guess the error bars are determined by the variation between cells. But I definitely think that perhaps also integrating the nascent transcripts, that at a certain point you can get rates. I think probably not at the single cell level, but you will have to average over these neighborhoods as we are doing in this analysis. But I think you definitely can get pretty quantitative here. The nice thing about the ribose-seq is also that it samples along the whole transcript. So it's actually more, in a sense, more sensitive than an RNA sequencing technique that's based on 3' detection, because we have much more RNA to sample from. So actually we see quite a bit of increase. We've seen an increase in sensitivity for certain genes. Most likely, the longer the gene is, the more sensitive we are. But I think, I think it can be very quantitative, but probably by averaging over a little, meta cells, so to say.
Great.
Thank you.
I think Graham has a question.
Yeah, this is Graham from Genentech. I guess it was actually kind of answered just now, but I was kind of curious about the coverage versus single-cell RNA-seq. If you get as many reads.
Yeah. Yeah, so it's very similar at the 3' end when you, for example, compare it to 10x chromium. But of course, we have access to the full length, so we will have more coverage than is, if you compare it to SmartSeq, it's pretty similar. Of course, if it's not translated, we don't see it, but that's the whole point. So this is, but yeah, so it's a full length sequencing technique. It's not based on poly-A enrichment.
Thank you. Very cool.
Great. Aviv has a question.
Alexander, that was beautiful. With the large number of genes, with the repression of the translation, sorry, my mask got stuck. Do you have a sense whether there's just kind of an overall cell biology disruption to translation during mitosis specifically, rather than an entirely regulated process? So for example, it could be that if you have a more structured UTR, you're less successfully translated, but because of some very global event in the cell that disrupts the ribosomes during all the events that happen to the nuclear envelope and so on.
Yeah, no, I think you're right. It's probably we have to search in that direction, because also we see this kind of really, black and white behavior between mitosis and not mitosis. So I think it's not something subtle. This should be something that affects many, many genes at the same time. What it is exactly, I'm not sure, but that we're digging into this.
Okay, so this reminds me of another thing. When you do nuclear sequencing with some of the protocols, you actually see it, right? The raise-and-seq protocol that we use, it has you can see the ribosomes basically in this, cobbled together with the ER in the nucleus. And so when you have a massive disruption to the nucleus in mitosis. So if you actually did single nucleus profiling using those kinds of protocols of the same cells, you might be able to see whether there is an agreement between these properties, and if so, then you might have this global effect.
Yeah, fantastic suggestion. No, I think that's very interesting.
Great.
Thank you.
One more question. Oh, sorry. There's an online question first. I apologize.
Hi there. We've got a question from Elaine Fuchs. What restarts translation after mitosis?
Yeah, so to be honest, this is also new biology for me, so I actually am not sure. So I don't know. Actually, we have to look into this. I have no answer to this question.
Okay, one last question.
Nicolas Fortelny from the University of Salzburg. I was wondering whether isoforms affect the comparison, because when in one case you look at the length and in one case you don't, because it's 3' or 3' or 5', and it switches to which RNA isoform or transcript is produced. Would this influence the comparison?
Yeah, so of course we have very short reads, right? So it's very difficult to really identify the isoforms unless you have very strong changes like exon skipping or something like that, or differential start sites. So it's hard from this data to really get very accurate information on this, because again, the reads are only 30 nucleotides long. So it's not that you can really span junctions, for example. But to be honest, we didn't look into great detail. Perhaps we might be able to have some resolution to actually distinguish between those, but it will be a tough experiment.
Great. Thank you so much, Alexander, for a wonderful talk.
Thank you.
Our next talk is by Mariana Baroni, and she's from the Brazilian National Cancer Institute.
Good afternoon, everyone. So first, I would like to thank the organizers for this invitation. It is an honor to be here today. And today I'm going to share with you some recent results from my group at the Brazilian National Cancer Institute. They are still unpublished. And in this work, we are trying to understand how these myeloid-derived cells, they behave in the tumor microenvironment and how they can foster the tumor growth. So the oncology field has benefited a lot from the recent advances regarding the characterization of immune cells in the tumor microenvironment. And as my colleague pointed in the last session, the immunotherapies, they have give many, how can I say, great options for many patients, but we still have a great proportion of patients that do not respond or acquire some resistance during the treatment. And there is many reasons why this happened, but one of them is because of the presence of some specific myeloid cells in the tumor microenvironment. So those cells, they are very heterogeneous. They can be derived from both adult or embryonic progenitors. They can also exhibit different polarization states, which allow them to exert different functions in the tumor microenvironment. So they are very interesting cells to study. And because they have been associated with many different functions in the tumor microenvironment, they are considering very interesting targets for anti-cancer therapy. However, there is a lack in our current understanding of like specific markers that can be associated with these specific cells, and we also still don't know in a deep, we still don't have a deep understanding of the roles of these specific cells. So thinking on that, we started this study by integrating data that was publicly available from 7 different solid tumors, also the normal tissue counterparts, comprising 142 donors and more than 400,000 cells. And with our approach, we were able to integrate data from 13 studies and to remove the batch effect associated with each of the studies we have used. And we were able also to integrate data from at least 3 different technologies. So after this integration, we selected the cells that we were most interested in, the myeloid-derived subpopulations that are circled here in this presentation. And then we started to characterize each of those cells using canonical markers that were already described in the literature. We later on dive deep into each subpopulation to also try to characterize some states they could present in the tumor microenvironment. So for conventional dendritic cells, for instance, we were able to characterize the type 1 CDC that were expressing CLEC9A, for instance, also a big cluster here in blue showing the type 2 dendritic cells that were monocyte-derived. Those cells were later on also subdivided in other cell subpopulations, reflecting 8 states. And we also were able to identify some mature cells as dendritic cells expressing LUMP3. Regarding the monocytes, we were able to identify the classical ones expressing CD14, also the non-classical ones expressing CD16. We identified one subpopulation that were expressing several markers associated with a very pro-inflammatory environment. We call them inflammatory monocytes, and also one subpopulation that were already expressing many macrophage markers. We also subdivide those subpopulations in some states that are depicted here in this picture, and we were also able to identify at least one subpopulation of monocyte that were exclusively found in tumor samples, expressing this heat shock protein A1A. Regarding the macrophages, we were able to distinguish between recruited subpopulations and that were expressing some monocytes markers and the resident one that were expressing markers associated with their ontogeny. And the literature has already shown that tumor-associated macrophages, they tend to express this receptor, TREM2. And we saw that many of the recruited subpopulations, in fact, they were expressing this marker, but we also saw a resident subpopulation that were expressing this molecule. And this resident subpopulation was also expressing high levels of the gene that codes for osteopontin, SBP1, which is also considered in the literature to induce the expression of PD-L1 in malignant cells, showing their immunosuppressive effect. We later on also subdivide those recruited cells in other subpopulations to study their states also of polarization. And regarding macrophages, the literature has characterized 2 main polarization states, M1, which is the classically activated which exhibit a more pro-inflammatory phenotype, and also this M2 signature, which exhibit a more anti-inflammatory phenotype. And by using genes associated with those signatures, we were trying to see if any of the subpopulations that we have identified, if they could be, if they were displaying more M1 signature or M2 signature. As you can see here for this figure, we have at least 2 populations that has a high score of this M1 signature. Also 1 population with a high score for this M2 signature. But when we look deep in these gene expressions, what we see is that none of those subpopulations, they clearly have the genes associated with those phenotypes all upregulated. So one subpopulation can either present markers for both signatures. So showing that this dichotomy that we use to classify macrophages, it's a very simple way of understanding their role in the tumor microenvironment. We also try to understand a little bit better about their role in the tumor microenvironment. So we investigated some genes associated with angiogenesis, and as we could see, these macrophages that were expressing high levels of IL-1R receptor N were enriched in this angiogenesis signature. We also investigated genes associated with invasion and metastasis, and we saw that this macrophage expressing high levels of metalloproteinase 9 were probably involved in this process. They were enriched with this signature. And we also saw that this macrophage expressing high levels of CCL7 was displaying several genes that were immunosuppressors, so showing their potential role in regulating other cells in the tumor microenvironment. We were able to validate those cells using an in silico approach. For that, we compared the subpopulations we have identified with populations that Mulders and colleagues have identified. We found some populations that were unique in our data set, but most of them we could correlate with those identified by them. We are now doing some immunohistochemistry experiments with some cohorts at the Brazilian National Cancer Institute, also to validate those cell populations. And last but not least, we try to identify the clinical impact of each subpopulation that we have characterized, and to do that we used the convolutional approach, where we were able to predict the abundance of each subpopulation in the tumor microenvironment of a bigger cohort of tumor samples, and in this case using bulk RNA-seq from the TCGA database. Here I'm showing just one example of ovarian cancer patients, and as you can see after this prediction, the tumor microenvironment of those patients, they are very, very heterogeneous. And when we associated this abundance of different cells with clinical parameters like overall survival, we can see that some populations, they are associated with this clinical outcome, as I mentioned, the overall survival. So in this case here for ovarian patients, we saw that the amount of macrophages displaying this signature, this CCL7, that has a very immunosuppressive signature, were associated with a worse outcome. This specific subpopulation was not associated with therapy response, but it was enriched in samples with a very low mutation burden. We were also able to predict and do the same analysis with different tumor types and subtypes. And here I'm summarizing some of the subpopulations that we found that showed some prognostic value in different cohorts. So the take-home message today is that tumor-associated macrophages, they have a very complex phenotype, and it's very hard to characterize them in those binary states, as the literature has tried to do. And this high-resolution characterization of those subpopulations is very, very important to detect new biomarkers and ways of, come up with new treatments, not only for cancer patients, but also for the diseases. With that, I would like to thank my group, also my collaborators from INCA, from Unicamp, UFSC, and the funding agencies. And thank you all for your attention.
Thank you so much for a wonderful talk. This is super exciting. I think we have some questions online. No? Chloe.
Congratulations, that's a beautiful data set. Immune cells are notoriously challenging to annotate, definitely macrophage as well. I'm curious if you could expand on how you define 13 stable macrophage states. How did you decide that these were distinct cell subsets?
Yes, right, thank you. Yeah, this is a great question because it's very hard to set a cutoff and say, okay, I have found the certain amount of cell populations or not. But what we were doing, we were investigating if we enhance or reduce the clusterization, if we were changing the variability among cell populations. So this was kind of an indication for us if the cluster we have was too variable or not. So this was an indication for us that we should, not going further, splitting the subpopulations, and we were also investigating the genes that were differentially expressed among states, if they were not making any meaning biologically speaking. We were like, no, I think we have done too much. So then we went back in this clusterization.
Thank you. I apologize if I forgot to introduce myself. So Chloe Flaney from Mass General Hospital and Broad Institute in Boston.
There's one question over there.
Yeah, adding on to that, in terms of the deconvolution that you're planning to do, does it make sense to retain the individuals to see how many subsets are shared across individuals as against defining clusters? Because by looking at common subsets, you might be missing unique populations, especially in the context of cancer, which might be quite important to the individual itself.
Yes, right. Because when we are analyzing single-cell data, sometimes we have a very small cohort and a small representation of the real heterogeneity that we can find, right? And when we do use the convolution analysis, what we can see is that the variability is really, really high. I try to show this presenting like the bar plots of ovarian tumors, and this happens with all the different tumors. So we have very distinct tumor microenvironment profiles in each cancer type we were studying. And this could be reflected in the survival analysis and the other clinical parameters. So it's a good way to correlate those subpopulations with those clinical data.
We have time for one last question, Aviv.
That was really beautiful, Mariana. I also have a question on the deconvolution, which maybe combines the previous two questions. So because the macrophages substates and the dendritic cell substates, they have a lot of overlaps. So for example, the way that you define your mregs for DCs and macrophage regs, they're based on, we could see the genes, similar genes, we all know that.
You're totally right.
How does it work for you in the deconvolution that you actually can reliably say it's this one versus that one? And is the degree of variation you see in the deconvolution similar to the variation that you see in single-cell profile tumors, so you're kind of confident that it's not, they're mixing each other's signal?
Yeah, this is a great question because depending of the matrix that I use as an input in the convolution analysis, I will have a different output. So we have tested several ways of doing this, and the way that for us was more reliable was to give an entire matrix with different cell types. So I have to give like malignant cells, T cells, dendritic cells, and so on. So in this way, I don't push the program to predict things that are not there. And another thing that we are doing is to use more than one methodology because there is this variation regardless the methodology we are using. And the third thing we are doing is to validate with immunohistochemic slides. So we really want to check whether those subpopulations, they are present and they have this impact on the clinical outcome.
Thank you.
Thank you so
Thank you so much. Wonderful talk. And the last talk for our session is from Jonathan Weissman. He's at HHMI and MIT at the Whitehead. This is going to be a pre-recorded talk. Jonathan won't be here to answer questions. However, at the end of the talk, he'll give us his email. So please feel free to just email him with questions.
I'm delighted to be able to talk today about our work on using CRISPR-Cas technology combined with single-cell readouts to follow the evolution of tumors in exquisite detail. All life, all cells, indeed all life, are related to each other. And as a consequence, we can organize cells or individuals into so-called family trees or phylogenetic trees. And this type of phylogenetic analysis is used very broadly in biology because it allows us to reconstruct the life history and infer critical events in sometimes distant past. This has become quite familiar, not only to the scientific community, but even more broadly with the SARS-CoV-2 pandemic, where by sequencing the virus at different locations and different times, it's been possible to trace the passage of the virus as well as its mutations to get, for example, then first the Delta and then more recently the Omicron variants, and to infer, before we knew anything about the molecular mechanism, that these two variants had increased in their ability to spread. Tumor, similarly, are an evolutionary process, and phylogenetic analysis of the evolution of tumors can address some of the most critical aspects of tumor growth and spread. So, for example, how do they start? How do they spread and metastasize? And how do tumors evade therapies? And this type of phylogenetic analysis has been used very broadly, both in model organisms and in tumors derived from patients to understand these processes. But until recently, our ability to reconstruct in-depth trees of tumor evolution have been fairly limited. This has changed with the development of CRISPR-based so-called molecular recording technologies that many labs, my own included, have been developing. Although there's a number of flavors of these types of approaches, each with their own strengths and limitations, the basic typical model for all of these is that you have a scratch pad in the DNA, so-called neutral region of DNA, which you can direct CRISPR-Cas9 to cut, for example, and to make insertions and deletions, or more recently, using things like base editors and prime editors, make point mutations or programmed insertions. These then, because they're in the DNA, get passed on to all the progeny cells. If there is an RNA copy of this DNA that's being made, then by pairing this with single cell readouts, you can read out not only the current state of the cell as evidenced by its RNA, but also the information that's been recorded. The key features of the approach that I'm going to talk about is that we have very high recording potential. In principle, we'd be able to uniquely mark every cell, even in a complex organism like a mouse. It's a continuous in vivo recording over months, and that, as I mentioned before, that you can simultaneously measure the cell lineage, so its history, and the current state through the transcriptome. So in a sort of a simplified form, the basic idea of this is we have multiple copies of these scratch pads, which enables us to record very large amounts of information that are pre-programmed into, for example, in work they'll talk about, a cancer cell. And then slowly over time, these scratch pads accrue marks, insertion, deletions, where here their location and the nature of the marks are indicated here. And when individual cells then get these marks, they then pass them on to all of their progeny cells, which then have the opportunity to accrue additional marks. Then at the end of the experiment, by RNA sequencing, you can get measure what all the indels or which locations that are marked and the nature of these marks. And from this, generate an allele table. So for each cell at each possible scratch pad, you know whether it was marked or not marked, and if it was marked, what the nature of the insertion and deletion was. And then these can be used through the principle of maximum parsimony, which is just a fancy way of saying that if two cells share the same mutation, it's more likely that they share because they had a common ancestor that where that insertion was, for example, was made rather than it occurred simultaneously, independently in the two different cells. So using this idea of maximum parsimony in our allele table, we can reconstruct phylogenetic trees. So this, of course, is a very simple one, but the trees that we can reconstruct are actually quite complex. This is an example of a phylogenetic tree reconstructed from a tumor that started with a single cell, had been growing in a mouse for about 2 months, and at the end consists of 5,600 or so cells. So each line here, each leaf here is a cell, and then it's a allele table. And then these are arranged based on this principle of maximum parsimony into the phylogenetic tree. And the kind of remarkable thing is that as you blow up this tree, you see that there's sort of fractal-like complexity relating cells that are most closely related to ones that are more distantly and even more distantly related to each other. And in this way, are able to really follow in enormous detail the evolution of the tumor. I'd like to then just end by talking about 2 applications that we've recently completed in the work using this approach. The first is for lineage tracing following metastasis with a xenograph of a cancer model. So here we would inject tumor cells into a lung cancer, tumor cells into a mouse, and then follow as that tumor grows and metastasizes to different sites. And through this, we were able to identify the roots of metastasis, sort of analogous to how the SARS-CoV-2 virus passage from one country to another. And we were also able to measure the rates of metastasis. And so how quickly, what was the intrinsic underlying rate, propensity of the tumor to move from one location into another. And then more recently, Jian Yang and Matt Jones in my lab, in a wonderful collaboration with Nir Yosef's lab and Tyler Jackson's lab, used this type of molecular recording technology to follow the very earliest stages of tumor development. So how a tumor went, we introduced in an adult mouse using Tyler's system 2 oncogenic mutations, a KRAS mutation in homozygous loss of the p53 tumor suppressor gene. And then over time, over the course of 4 or 5 months, the tumor would evolve to perform an aggressive cancer that extravagates and then ultimately metastasizes. And what we were able to do is to reveal a hierarchical, the hierarchical nature of the process by which this happens, which we see that there's the tumor starts in a relatively normal cell state, despite the oncogenic mutations, then goes through a period of very high plasticity in which the cells are able to rapidly jumping back and forth between different cellular states. And then eventually one of these states wins out and further evolves to become to allow a selective sweep of a rare subclone, really analogous to, for example, what Delta or the Omicron variants did. And only from these high fitness, low plasticity state, then do the metastases arise. So just stepping back a bit, we more generally, I think this type of technology is going to be a critical addition to our to these types of cell atlases, in which you can use, for example, Cas9 or even the natural mutational process and work I don't have a chance to talk about with Vijay Sangran. We've been using natural mutations in mitochondrial DNA to follow, to create phylogenetic trees from human samples. These then allow you to record information about the history of the organism in its DNA, and then to read it out at the end of this experiment, much the way that same way analogous to how a flight recorder allows you to understand how an accident happened by giving information about the processes that occurred before that accident. So just finish by thanking the people who did this work. I've had the pleasure of working with a number of great trainees and colleagues, but just give a shout out to a few of them: Michelle Chan and Zach Smith, and a wonderful collaboration with Alex Meisner's lab, just some original recording work following mouse development. Jeff Quinn and Matt Jones, a terrific collaboration with Trevor Bavona and Nir Yosef did the metastasis work, and Jian Yang and Matt Jones with Nir and Tyler Jacks did the work on the KP model of early evolution of tumors. That I'll thank you and
well, I guess thank you for Jonathan for a great talk. And if you need his email address, maybe Christine can give it to you. And if you have additional questions, both Alex Marson and Nir Yosef are here at the meeting, so you can ask them as well. They were acknowledged in the last slide. So let's thank the speakers for really exciting session. And since there were quite a few questions asked online, very general questions, Jenny is going to, or Sam is going to ask some of these questions, and Aviv will answer. Or anyone else can answer as well.
Oh.
A microphone.
I don't see it.
What's the question?
We need a second.
We have a shortage of microphones.
Thank you. Yeah, so we've had a couple of questions come in from our virtual attendees about the scope of the Human Cell Atlas and multiomics. And the first one actually was from this morning from Fatma Betul Discanlan from iHealthTech in the US, asking about the importance of secretome, especially from the human protein, or sorry, from the proteinatlas.org, and wondering about how HCA works with this. And the second one...
maybe I'll answer the first one first.
Ask the first
on the secretome, and I'll start, and maybe Sarah or others would want to answer. I'll consider them, Sarah, as though these were questions in the morning when we gave our talk, because it's not really clear. So I think with the secretome, there's great opportunities, especially in spatial data. But especially in spatial data that is multi-omics, because what is interesting about the secretome is that, of course, you want to know the source of what is being secreted, but you also want to know where it is. So the source, you need something that stays inside the cell, and that can be the RNA, and where it ends up being, where it's secreted, would be measured at the protein level. And so that is something that in an in situ assay, you get kind of the perfect answer in a spatial assay. You get the source in the RNA, and you get where the protein is. Now, I want to say, because Alexander talked later on about the translatome, and maybe that also engaged the person asking the question. When you look at protein translation, you can look actually at protein translation, and when you also do actual protein measurements, not just for the RNA, then secretion looks the same as degradation. The proteins disappear from the cell. So again, it's a way, if you're seeing something getting translated, but the protein is not there, it is a way of inferring things that are being secreted. So I think there's great opportunities for the secretome. I don't know, Sarah, do you want to add to that?
No, yeah, I think as we said, in tissue sections in vivo, it'd be great to see how far proteins diffuse from the source cell and to cells that have the cognate receptors. And I don't know whether the person is referring to in vitro secretomics, but that's also a fantastic way of characterizing the phenotype of cells, immune cells or other cells actually in general. So a great complementary data for cell phenotype in the human cell atlas.
And the second question came from Giorgio Catoretti for the University di Milano, Bioca, talking about in situ proteomics by MALDI-MS, saying it's approaching single-cell resolution. And can we explain why? Is HCA using this, and if not, why not?
So so deep visual proteomics and those kinds of technologies will be super interesting and exciting, I think, to integrate with the spatial methods by sequencing, by imaging, and then also the single-cell proteomics. Did you want to say also, Priska?
I'll add two things. First of all, in the overview we gave in the morning, we actually said there's approaches of different kinds, and we highlighted a couple, but there's just a long list of them right now. This is because there's many advances. And so this is one of them, and generally, things are going very well these days for single-cell proteomics, as well as for spatial proteomics at single-cell and subcellular resolution. So we're going to see a lot more of that. Just to extrapolate from past patterns, first the methods mature, then they become better disseminated, then you see the biology that everyone does with them. So I would predict the same for this one. I will point out that in HubMap, which is one of the major initiatives, this is one that's funded by the NIH, that focuses on building cell atlases, there has been, I believe, MALDI-based approaches for quite a while. Kati might remember a little more exactly, precisely where they stand right now, but there's definitely been work around that in some tissues. I don't know if others want to comment on that. Seems satisfying.
Thank you.
Any additional questions?
Priska said lipidomics next. There's actually really nice work from Sean Davidson. Yeah.
You should say.
Sorry. Yeah. No, there was a very nice work on single-cell lipidomics of D'Angelo and Lomano at the EPFL, so I think that would be.
Yeah, and there's now spatial lipidomics. There's a paper in Nature Methods from a few months ago from Sean Davidson in Princeton. So again, these things are coming. If you know it or not, you should get ready. And then Fabian and Dana can figure out the rest.
Great. Thank you so much for answering all these questions, and hopefully we'll have a lot more questions and discussion tomorrow as well. And Christoph is going to end the day with final remarks and some housekeeping notes.
So thank you for staying all to the end, but this is not the end yet. Today we had we heard a lot about exciting science, and I think this brainstorming was already, now this last question and answers is already what's the topic also for the after-hour discussions, like what's next, where should we be going, what is exciting new. Tomorrow then, we will have, is this day 2? Exactly. Tomorrow, things become more concrete. Tomorrow is all about how we are going to use the data of the cell atlas and how we can make it accessible. So whoever got excited today, also of the people who are watching us online, about all the cool things that can be done with single-cell data, tomorrow is the opportunity to break this down to reality with dedicated, like a morning session or starting 9 o'clock Vienna time, discussing the opportunities and challenges using the data, and then in later in the day, a breakout session on this precise same topic. Also the biological network sessions, that's also a great opportunity to get involved in the discussions. And of course the keynote lecture in the early afternoon by Elaine Fuchs that we're all looking very much forward to. And tomorrow 7 o'clock, for those people who are on site, tomorrow 7 o'clock in the evening, the conference dinner. So that is for tomorrow. One more thing really for tomorrow regarding COVID. So kind of statistically, 1 or 2 of you will develop COVID tonight. And it would be great if that person realizes that and stays at home and watches us virtually, because we don't want to really make this a super spreading experiment here. I know that's hard because of all the cool science, cool people, and interesting discussions, but yes, you can do it. So if you have symptoms, watch us online, and that's great. We will be forever thankful for that. Obviously, if you have no symptoms and you're as fit as ever, be here 9 o'clock latest and join all the discussions. Good. For today, we have 2 more things. We have the reception afterwards, food and drinks. I hear there's some wine, but also non-alcoholic beverages. But before that, there's one more duty that has to be done with a smile, which is the group picture. So this is our dear photographer who will now steer us all into place, and it's really difficult to get all the people here, plus the ceiling and the chandeliers, etc. But we will manage somehow, and I'm sure it will be great, and you will remember forever after. So come, please, for the group photo.